Good morning, everyone. Um, Welcome to the second to last week of biochemistry. For some of you, the second to last week of your undergraduate careers. You're so close. It is somewhere between March and August. I no longer know or care. Let's get started, shall we? Today we'll be talking about the mechanisms of G protein coupled receptors and secondary messengers in more detail. Before we delve into some of the mechanistic details of this pathway, I wanted to make a general analogy. Have you ever seen the movie Ex Machina or heard about it? In case you're binge watching movies, this is a pretty good one. It's kind of disturbing, but it's this movie, it's this indie sci-fi movie, and it deals with this robot. Her name is Ava, and it really is dealing with the idea of artificial intelligence, which is a very, very basic definition, the ability of a computer program or machine to think and learn, to respond. In this movie, you have, in addition to Ava, two other characters. You have Nathan, who's the creator of Ava, and he invites one of his employees, Caleb, on this weird, strange retreat, and he doesn't know really what's going to happen, but he ends up being a means to test whether or not this machine truly has artificial intelligence. If you're a real nerd, you may have heard of the Turing test. Anyways, it's a really cool movie, but that's the premise of the movie, and things get out of control. There's one point in the movie where Caleb is asking Nathan, how he came to create Ava, and they walk into this one room, and behind Nathan is this painting, and it's actually, I did a little research on this, and I thought you might find this interesting. Just trying to find ways to brighten your, social distancing by making it a little interesting. So this painting is actually by a guy named Jackson Pollock. And there's this one cool quote in the movie where he says, "The challenge is not to act automatically." And he's talking about this painting that's behind him, and this notion of out of chaos, you get order. This is a picture of one of Jackson Pollock's most famous paintings. It's called Convergence. It's really cool in that, you just look at it and it's just fun to look at. It's also huge. It's roughly 8 feet by 13 feet. So this is a massive painting. And this artist is actually someone who experimented with something called abstract expressionism. I won't get into the details of it, but he uses this creative process. I don't even know how to pronounce that word, Janusian. Anyways, it's kind of an interesting story because he used this process of a new way of doing art to help him overcome bipolar symptoms, which is really interesting. But the idea here is that you have chaos. And as you're creating this sort of connected tapestry of something, it's not just chaos. There's also order within that chaos. And you, what defines chaos, I would say, is just randomness, right? It's you act automatically without really any direction. But in order, like you, it's not automatic. It's in a coordinated way. And so the review that you read at the very beginning of the semester, I'm trying to bring it somewhat full circle here, where you read a review article by Bruce Alberts, how the cell was comprised of a collection of protein machineries, the proteins that we've spent all semester talking about. And that really is what's going on here. Whether we're talking about coagulation cascade pathways or GPCR secondary messenger systems, we're talking about the controlled flow of cellular information, which is one of the core concepts that we also talked about at the beginning of the semester. There's this really fascinating review article if you're interested in reading more about it, but this is talking about how scaffold proteins actually serve as ways to modulate the pathway. In last lecture, we talked about the idea that not all parts, not all macromolecular components of a signaling pathway have enzyme activity. Some of them just help modulate who binds to what. So that's what this review article is about. What is the logic underlying these signaling pathways? It's what we talked about last lecture. You can boil down the essence of how these pathways are put together by these themes of sensitivity, specificity, modularity of the signaling proteins, desensitization and adaptation, integration, and localized response. So the goal for today is to get into the nitty-gritty details as much as possible for G protein coupled receptor mediated pathways, in particular using the model G protein coupled receptor, which is beta adrenergic receptor. One question I'd like you to be able to address, it's more conceptual, but A, what is the function of GPCRs, G protein coupled receptors, and secondary messengers? And how is it different from the coagulation cascade that we studied? Well, I think the simplest definition of a G protein coupled receptor and secondary messenger system is that it's transducing an extracellular stimuli to an intracellular signal. And this really gets at a key difference between what we're about to talk about and coagulation, which is the presence of a membrane, right? In coagulation, where we had fibrinogen being cleaved into fibrin and then forming a gel-like substance, this was all happening outside of the cell. There was no need to transduce a signal from outside a cell into a cell. That's not to say that there aren't other signaling pathways involved in addition to the aggregation of fibrin, right? I mentioned the idea of there are definitely signaling pathways where you cross a membrane of a platelet, for example, that can then have an effect on recruiting components of the immune system, for example. But coagulation, at its most fundamental level, doesn't need a cell. So that's the key difference. In coagulation, we had an extracellular stimuli and an extracellular signal. So the signal need not move across a membrane, and that's why in the textbook, it doesn't really count coagulation as a true signaling pathway because you're not crossing a membrane. But it follows the same logic. All of these themes are also true. So now let's get into it. I'm trying my best to give you some cues as to what to focus on. Here are a few of them as we go through the next few slides. I'd like you to be able to identify 3 components involved in signal transduction through G protein coupled receptors, distinguish between primary and secondary messengers, recall the effects of secondary messengers on downstream components of pathway. So what do secondary messengers do? Do they positively and or negatively regulate things that come later in the pathway? And then from a human disease perspective, describe the diversity of G protein coupled receptors that are encoded by the human genome, have a general appreciation for the total number of G protein coupled receptors. It's astonishing. Be able to describe some of their physiological roles. It's kind of a wash because it's almost everything. It's hard to find something that they're not involved in. And then be able to describe how mutations in this pathway can cause disease. First, I'll discuss it generally, but similarly to the in-class activity for last week, dealing with mutations in parts of the coagulation pathway, there will be a series of questions that ask you, okay, there's a mutation in, say, the G protein that causes its hydrolysis of GTP to be modulated. What happens? And then lastly, just generally be able to explain why G protein coupled receptors are such good drug targets. So this is figure 12.4 in section 12.2 of your textbook. Again, this is looking at the beta adrenergic receptor pathway. Just be aware that there are many different types of G protein coupled receptors, not just beta adrenergic. And while they fundamentally are the same, they can act in very different ways. So what 3 components would be the same in all of these signal transduction pathways involving G protein coupled receptors? The 3 components are, first, you have a plasma membrane receptor that has 7 transmembrane helical domains. As the name implies, G protein coupled, the second component is a G protein. By G protein, the G stands for a guanosine binding protein. So a guanosine nucleotide binding protein. So GDP and GTP are nucleotides, and the G proteins bind to them. And depending on what's bound to them, that will determine if it's in an inactive or active state, because there are conformational changes involved with either being bound to GDP or GTP. The third component is an effector protein. Here in the beta adrenergic receptor pathway, that effector protein is adenylyl cyclase. Excuse me. Having a little bit of technical difficulties here. God damn it. I don't have time for this. So let's take a look at figure 12.4 in your textbook. This is section 12.2. And a reminder that we're only talking about one specific G protein coupled receptor, the beta adrenergic receptor. Just know that there are many different G protein coupled receptors. They don't act necessarily in the same way. But they do have shared themes. They have the same 3 components that will always be involved, regardless of what GPCR you're talking about. So What are the three components of G protein coupled receptors? The first is the G protein coupled receptor itself. It is a plasma membrane protein, so it's an integral membrane protein, and it serves as the receptor. It has 7 transmembrane helical domains, and it is responsible for binding of either an agonist or an antagonist. The agonist in this case being epinephrine. The second component, as GPCR, the name implies, G protein coupled, is the G protein itself. The G in G protein stands for a guanosine nucleotide binding protein. So it's binding to nucleotides that have guanosine, either GEP or GTP, diphosphate or triphosphate. And depending on what's bound, it will either be in a so-called inactive or active state. This is because GDP versus GTP bound causes a conformational change in G protein in this particular subunit of the G protein. We'll get to the subunits in a second. But it impacts what it binds with and is subsequently activating. The third component of the pathway is an effector protein. Here for beta adrenergic receptor pathways, that effector protein is an enzyme called adenylyl cyclase. The enzyme activity involves converting ATP into cyclic AMP. It's important to note that not all effector proteins are enzymes. Some are ion channels that just allow things to come in and come out. And that's what activates secondary messengers. So there's our three components: your GPCR, the G protein, and the effector. How about primary and secondary messengers? So the primary messenger in this case is going to be considered the agonist or antagonist that's binding. So here it would be epinephrine binding to a cavity in the extracellular face of the G protein coupled receptor. That's your primary messenger. Secondary messengers are on the inside, in the cytoplasmic side of the cell. And so here the secondary messenger is cyclic AMP. The secondary messengers, by definition, are low molecular weight metabolites or inorganic ions. So these are not big, massive proteins. So what gives this particular pathway specificity? It has to do with the receptor. If you remember back to the previous lecture, when we talked about specificity versus sensitivity, we talked about like developing a coronavirus diagnostic test. Specificity had to do with excluding certain things as opposed to being very sensitive but recognize. Okay, so if you remember back to last week, we talked about the difference between specificity and sensitivity. Specificity has to do with binding only certain things and excluding the binding of others. In the context of signal transduction pathways, specificity is achieved by what two things? Molecular complementarity, one, and also two, expression of genes. So in the context of GPCR signal transduction pathways, specificity is achieved primarily through, not surprisingly, the first part of the signal recognition, the receptor. So specificity, there are different isozymes. You can have alpha-1, alpha-2, beta-1, or beta-2. These are all adrenergic receptors, but they bind to slightly different ligands. So alpha-1 and alpha-2 may not bind to epinephrine, but beta-1 and beta-2 do. So that's one level of specificity that we're obtaining in this pathway. The second is differential expression of these isozymes in different cell types. So not only in the context of a single cell, but a multicellular organism, you don't want, another way to have specificity of signaling is only express it where you need it. What was the biological process we were mediating here that I said? This was the binding of epinephrine to modulate a fight or flight response. And here we're talking only about beta 1, beta 2. So which cell or tissue types is it expressed in? Again, you can have all of the cells in your body. Hopefully you remember this hopefully this is something you've learned like in cell biology or genetics. All of the cells in your body have essentially the same genes, the same genetic information, but they are so different because different genes are expressed differently. This is true also for the genes encoding the beta 1 and beta 2 adrenergic G protein coupled receptors. They are preferentially expressed in muscle cells, adipose cells, and liver cells, specifically a type of epithelial cell associated with liver tissue called hepatocytes. So again, this has to do with gene expression, going from DNA to RNA, RNA to protein. Not all genes are expressed all the time, and some genes are never or rarely ever expressed in certain cell types. So how many G protein coupled receptors are there? As of now, we know that there are roughly 800, a little more than 800, which is a lot, that are encoded in the human genome. Roughly 350 detect hormones or growth factors, and the other 500 are involved in olfactory, which is smell, or gustatory taste receptors. The biological process being mediated here, we haven't really gotten to it yet, but the final result of all of this signal transduction pathway, last week we talked about coagulation. The final end product was fibrinogen glomming on together to make a clot. Here, it's actually going to cause a release of glycogen, which can be used as energy. This is what the fight or flight response is kind of all about. If you need to run, you need energy to run. So glycogen is an energy source. So if you're being hunted by a velociraptor in Jurassic Park, well, epinephrine is going to bind to the beta adrenergic receptors in these cell types of your body. But again, you want it to be specific. It's all about sort of conserving energy. If all of the cells in your body had these receptors, you'd be wasting a lot of energy, releasing glycogen in places where metabolism couldn't occur. Okay, so let's walk step by step through the beta adrenergic pathway. The first thing to point out is step 1. Again, we have epinephrine binding to the receptor. It's sort of, if you were to picture it, it sort of has like a hole in the top. The GPCR does, beta adrenergic receptor. The epinephrine binds into a cleft sort of deep even within the plasma membrane. You can see it's in the plasma membrane here, and it causes a conformational change that is transduced all the way to the other side of the beta adrenergic receptor. So it promotes a conformational change in the receptor's intracellular domain that then affects its interactions with an associated G protein. So this conformational change is being transduced to G alpha or GS alpha. We have to take a little bit closer look at the G proteins. There are heterotrimers. Here's the alpha subunit, beta subunit, and GABA subunit. Which subunit binds to guanine nucleotides? It's the alpha subunit. This leads to the first step is propagated to a conformational change in the alpha subunit. This is significant because this conformational change favors the binding of GTP versus GDP. So in step 2, GDP dissociates, and there's lots of GTP inside of the cell, and it gets replaced by that. So that's step 2, where you have exchange of your guanine nucleotide. This is what is meant by, this constitutes a molecular switch. And these are really critical. When GDP is bound, it's in the off state in terms of promoting further signaling in the pathway, and in GTP bound form, it's on. So here's a closer look at that molecular switch, figure 12-5. This is showing you that GS with GDP bound is turned off. What do we mean by turned off? That it can't interact with the effector protein. So interactions between alpha and beta gamma, if these things are interacting, alpha can't interact with adenylyl cyclase. So again, it's binding of the hormone epinephrine that first causes release of GDP and second causes replacement by GTP. So when GTP is bound, alpha dissociates from beta gamma. So we're no longer heterotrimer, but beta and gamma actually stay associated with each other as a dimer, but then alpha can go and touch through weak interactions adenylyl cyclase. This is now on because, again, it's all about propagation of conformational changes. Now you have a conformational change in adenylyl cyclase such that it's catalytically active. So what causes the alpha subunit to dissociate from the beta gamma subunits within the G protein? The answer is epinephrine binding to the G protein coupled receptor, and then a conformational change being propagated to the alpha subunit. So this is where they're transitioning from a heterotrimer in the GDP bound state to a dimer, beta gamma and a monomer alpha, that is then replaced by GTP. These cartoons are sort of informative, but they don't really do a good job of highlighting the fact that when GTP bound, there was a new surface revealed. There's an allosteric effect whereby a new surface in the alpha subunit is now accessible that can then touch adenylyl cyclase. What are those? Just interesting note, you see that some of the parts of the G protein kind of go into the membrane. So it's important that all of these components stay tightly associated with the membrane. If they didn't, they would just diffuse in the cytoplasm and wouldn't be able to interact with adenylyl cyclase frequently enough. So parts of certain subunits of the G protein, such as alpha and gamma, are covalently modified. There's a palmitoyl group that allows it to be inserted basically always associated with the membrane. So I might ask you to distinguish what are of the 3 components of G protein coupled receptors, which are integral membrane proteins. Those would be the GPCR itself as well as the effector, and which are not. Which are not integral membrane proteins, that would be the G proteins. But then how are they still associated with lipids? Palmitylation. So the G alpha subunit or GS alpha subunit is held to the membrane by a covalently attached palmitoyl group. And this is sort of a theme that's used by the cell over and over again, adding what I like to think of as lipid shoes. These are post-translational modifications, lipid shoes that are added to some protein, and it allows that protein to walk to localize to a specific membrane. If you're interested in reading a little bit more about the different types of lipid post-translational modifications, there's this really good website that I found here by William Christie. Here you can see a palmitylation involves adding this really hydrophobic chain to a cysteine residue of, this would be of a side chain on GS alpha. But you can also add to different side chains such as glycine and have a similar molecule, or add slightly different molecules that are, they have some double bonds. They're, what would that be? Unsaturated. But they still have the same effect of causing whatever you've added this to be added to lipids. So this discusses palmitylation. And we're still in section 12.2. But this also talks about farnesylation, which is just an interesting link. Again, I'm trying to tie this all together for you through the progeria assignment. Palmitylation is similar to farnesylation in that it has the same impact of recruiting a protein to a membrane. In the context of progeria, we were talking about post-translationally modifying a lamin protein that caused it to be recruited to the nuclear envelope. But here we're actually dealing with recruitment to the plasma membrane. All right, so now that we have a better understanding of how certain parts of the G protein, the GS, are staying localized to this area where a signal is being produced, let's continue on. We left off at step, I believe, 2, which is that the binding of epinephrine caused a conformational change that was propagated allosterically to the GS alpha subunit that allowed it to dissociate. Step 3 from beta gamma. Step 3 is that after the beta and gamma subunits of GS dissociate from alpha as a beta gamma dimer, the GS alpha exchanges GDP for GTP and moves within this plane of the membrane. Because of the palmityl group, it's able to stay associated to a nearby adenylyl cyclase effector protein and then activates it. So that's step 4. Association of GS alpha with ATP to the adenylyl cyclase now stimulates adenylyl cyclase enzyme activity, and you have production of your secondary messenger. So at this step, we've actually covered a few additional themes, the logic of signal transduction pathways, but they may not be completely clear to you, so let's walk through them. Modularity of the signal proteins. It's everywhere. The most obvious example is probably the GS alpha subunit, right? It just looks like a green weird booger blob, but it has different functional domains. If you were to actually look at its structure, it's modular. It's binding to at least 3 different things you can see. So one chunk of it is binding to the GPCR, the beta adrenergic GPCR receptor. It's also a separate domain is binding guanine nucleotides, and then even there's another modular component to it that is binding the effector adenylyl cyclase. So that's modularity. Sensitivity. So this is where you start to get, it's kind of like a pyramid scheme where you activate, one activates 2 things, those 2 things activate 4 things, and geometrically the signal just takes off. So sensitivity, examples of sensitivity could be, for example, once you have a GS alpha subunit that is bound to ATP, that can actually bind multiple adenylyl cyclase effector proteins. Shown here for simplicity, there's just one, but in the cell there might be many, 10, 20, 50, 100 adenylyl cyclases that can be activated by a single GS alpha subunit, as long as the ATP is bound to it. You can then go even further, right? The adenylyl cyclase itself is making many cyclic AMPs, and many cyclic AMPs are activating many, we haven't talked about it yet, but it's the next step is the secondary messenger is going to activate a protein kinase A. So that's the sensitivity aspect of it. At this point, you might be asking yourself, okay, time out. Similar to the blood coagulation cascade pathway, you had tissue factor that was activating 7A. Remember your first protease? But then the idea was, great, we have a blood clot forming, but what if it were just to run away? What prevents runaway signaling? In the blood coagulation pathway, it was protease inhibitors, primarily, crap, I forget, trypsin inhibitor? No. Whatever, you can look back. Here it's interesting. Have you seen any proteases in this pathway yet? Nope. Right now it's just non-covalent interactions. There's no making or breaking of covalent bonds. It's just proteins non-covalently interacting with each other. So what prevents runaway GPCR signaling at this point? Sorry, I'm high on the movies. I went on a rom-com binge. I didn't watch Runaway Bride. I really like Julia Roberts. I did watch Notting Hill. That's a really good movie, but I'm old. It's kind of cheesy. What prevents runaway GPCR signaling at this point? So there are at least two things that are preventing runaway signaling. One of them you can actually see if you were to peek at the last part of the pathway. Do you see which of these options might dampen the signal? Well, we'll come back to that. The more interesting one, at least from a biochemical perspective, is that the G alpha subunit is like a timer. Okay, what do I mean by that? It binds ATP, right? And when it binds ATP, it can activate the effector protein. But what you may not appreciate is that as soon as GDP is exchanged and now GTP is bound, this GS alpha subunit is actually an enzyme that hydrolyzes GTP back to GDP, right? So as soon as you've activated this GS alpha subunit through binding of GTP, it's at some rate, it's going to be self-limiting. It's going to hydrolyze the GTP back to GDP. What would happen then? If you had GDP bound to the GS alpha subunit, it would no longer be able to bind to the effector protein and stimulate the production of your secondary messenger. And instead, actually, GS alpha would then go back to binding to the dimer of beta gamma and be sequestered away and be ready for maybe a new signal. So that's part one. This timer is trying to highlight that at some rate, GTP will be hydrolyzed. So it's not like this is forever going to be it being GS alpha stimulating the pathway. The second answer, if we peeked ahead, is that the secondary messenger itself is degraded. By what? I don't know. We don't need to know. But it's actively degraded, right? So cyclic AMP, there's something that comes along and starts breaking a covalent bond, or many, I don't know. But the secondary messenger itself is degraded. The secondary messenger in this case is always activating the next step, which is activating the activity of a serine-threonine kinase. So this is PKA, protein kinase A. Protein kinase A then phosphorylates all these different targets that has an effect ultimately of releasing glycogen. So let's take a closer look at how cyclic AMP is allosterically activating pKa. So in purple here in step 5. So closer look at allosteric activation by pKa by cyclic AMP. So this is a kind of a confusing diagram, but it actually tells kind of a cool story. And it has a good conceptual link to what we saw in coagulation. So pKa is comprised of 2 identical catalytic subunits in C. So those are these blue guys here, as well as 2 identical regulatory subunits shown here in red. Initially, it forms this tetramer. It dimerizes through this dimerization domain in the regulatory protein. But the key here is that in this tetrameric form R2C2, notice where the domain of a regulatory subunit is occupying the catalytic subunit. So it's binding to a substrate binding cleft of the catalytic subunit. So if we come back up here, like in this diagram, you can see it kind of looks like a butt or something, or whatever, some sort of cleft. The regulatory subunit is sitting there. And I think that's what's shown here in the actual 3D structure, which is that you have your regulatory subunit, and you can see there's this nice pocket in the catalytic subunit. This is, remember, pKa is a serine threonine kinase. It's going to phosphorylate substrates, but the substrates need to sit here in order to be phosphorylated. So in this state, can pKa phosphorylate downstream substrates, like the thing that initiates glycogen secretion, phosphorylase B kinase? The answer is no. So it cannot. The R subunit is blocking interactions between pKa-C subunit and its substrates. This is like, again, I don't know what else analogies to use. This is like, you know, you don't go by yourself when you go out partying. You don't want to just be accessible, right? So your co-wing, your wingman or woman or wing molecule, in this case, the R subunit, make sure that you don't get crazy until the right time or with the right person. They've got your back. So, yeah, in this state, okay, I already said it. This is autoinhibition. So then if you look at this diagram, and I already said the answer, what relieves the autoinhibition? You can see here, now the cute little catalytic subunits are free. What freed them? Cyclic AMP binding. So cyclic AMP binds, there's 2 that bind to each of the regulatory subunits and cause a change in its shape such that it can no longer interact with the C subunit. Now these guys can go and phosphorylate things like phosphorylase B. It gets really confusing, the terminology, but this is a kinase that is phosphorylating another kinase that is confusingly named phosphorylase B. And phosphorylase B, I believe, then phosphorylates something else that results in release of your glycogen. It's quite amazing that this is all happening very rapidly, similar to the blood clotting mechanism. So what relieves autoinhibition? Binding of cyclic AMP. In terms of the coagulation pathway, does this make sense to you? Like, you don't want the pathway to be on all the time. You want it to be on only in response to a signal. So what is this analogous to in the coagulation pathway? In the coagulation pathway, we didn't really talk about any kinases. They were all proteases, right? They were all proteases that were initially inactive. as a what? And then active following what? Starts with the Z. Good. They were initially inactive as zymogens in the coagulation pathway, and then active following cleavage by another protease. So conceptually, it's the same idea. Start off inactive, then become activated. But in terms of the mechanism of autoinhibition, it's completely different. Here, we didn't have a cleavage of anything. And as a result, here you can actually reuse PKA, protein kinase A, right? In the sense that PKA can go back to being turned off. PKA is turned on here, but whenever these two cyclic AMP molecules let go of the regulatory subunit, it can glom back on here. The C subunit can be autoinhibited once again. This is because, right, it does not involve cleavage, but just an allosteric change leading to alternative non-covalent binding, which is a mouthful, but hopefully you get it. For the proteases, once you converted them to an active form by cleaving them from the zymogen to the active form, they're always on. So something has to happen. And there are things that happen that we don't have time to cover, but I'm just trying my best to show you similarities between these two pathways. Okay, we're almost done. We're heading down the home stretch of this particular pathway. So hang in there. There's one more layer of detail that I'd like you to be able to know. And by being able to know this, it gives you the ability to predict how mutations in components of the pathway are involved in human disease. Similar to last week's in-class activity, I'd ask questions about, a mutation in this component would cause what effect. But there's two additional components to the pathway that we haven't talked about that deal with having to take a closer look at the G proteins and how they function. The two additional factors are GEFs and GAPs. Let me walk you through this. Let's take a closer look at G proteins. I would highly recommend reading box 12-1 in detail. It even walks you through some examples of mutations in some of these components that I'm about to talk about. For the final exam, I'll probably ask a few questions that are very similar to the questions that are discussed in box 12-1. Box 12-1 is dealing with, it's talking about other G proteins, not just those involved in beta-adrenergic GPCR signaling, and how they are involved in health and disease. So previously I talked to you about how the G protein is like a switch, either on or off, whether it's bound to GDP off or GTP on. And there's a secondary aspect of how it's also a timer, because once GTP binds, at some rate, the G protein will hydrolyze it back to GDP. So both of these additional modulators, GEFs and GAPs, impact the timer aspect of it. Well, and the switch aspect of it. So remember how many G protein coupled receptors there were? There was something like 800 or so that were known. There's also lots of G proteins, the things that are coupled to the G protein coupled receptors. So the human genome encodes roughly 200 G proteins, and all of them share the common feature of nucleotide hydrolysis. This is what, again, is causing the G protein to act like a molecular timer. This is kind of like if you have a fancy bathroom or at least if you go to a hotel, you know how you set the timer when you take a shower to get rid of the steam? Eventually it goes off. That's kind of what a G protein is doing. This shared function is ultimately provided by shared structure, right? So structure equals function. To the left is shown a picture of another G protein called RAS. And you can see that, remember how I said when GTP binds, there's some magical conformational change that reveals something that can then bind to an effector. This is showing you where in the structures those relevant parts are that can now interact with effector proteins. So in the case of RAS, there are two switch regions that are initially buried in the core of the protein when it's bound to GDP, but become accessible when GTP is bound. So you can see you have this really cool looking structure. It's got some alpha helices and some beta sheets. But here in here's switch 1 in this sort of flexible region here. And where's loop, switch 2 is in green, this alpha helix here. And you have GTP or a GTP analog to get this structure bound in this region here. The structural motif that is key to GTP binding and subsequent conformational changes is something called a P loop, which is in this region right here. And there are two residues that are particularly important for whether or not you're in a conformation that the G protein can bind to effectors or not. So the P loop is very important because it is what binds to GTP. This next figure in this section is showing you schematically what's happening. Here you have RAS, and the big sort of dome thing is in blue is the P loop. Okay. So in RAS, the gamma phosphate of GTP, here's GTP. You have guanine, a ribose sugar, and 3 phosphate residues. The last phosphate, or the gamma phosphate of GTP, is binding to a few things. First, a lysine residue in the P loop. It's not shown in this figure, but also 2 key residues, threonine and glycine, which were here in the structure, really close to where the GTP is, as well as here, which is also kind of close. You can see how they're both in the region of this gamma phosphate of GTP. Okay, so the gamma phosphate has lots of oxygens, and so this is one instance where I think it'd be nice to go dig. This is probably as deep as we'll dig in terms of like structural binding and how it's involved in GPCR signaling. But the oxygens hydrogen bond with these residues, either threonine or glycine. So when GTP is hydrolyzed by the intrinsic GTPase activity of your G protein, you have loss of hydrogen bonds to threonine and glycine, threonine 35 and glycine 60. So these spring-like things are the hydrogen bonds. So when you cut this off, right, you would now go from GTP to GDP. You would no longer have hydrogen bonds. When you no longer have hydrogen bonds, these guys there'd be a conformational change, and it goes to a relaxed conformation where the G protein can't interact with effectors anymore. So let's go back, because we hopefully are trying to get in a frame of mind of like preparing for the final as well. let's go back and think about what are glycine and threonine. So if I were to ask you this question, which parts of the G protein, these residues of the G protein are involved in hydrogen bonds with the gamma phosphate of GTP? So here I'm showing you the structures of glycine and threonine. What chemical characteristic would you have to interact with a negatively charged oxygen? You would have to have some polar characteristic, right? Which of these is polar? One, both, or none? So glycine, its R group is nonpolar, right? It's just a hydrogen. Whereas in threonine, you do have a polar group here that could potentially, this alcohol group could potentially hydrogen bond with, the hydrogen of this alcohol group could hydrogen bond with the oxygen. Sorry, this is a lot to cover, so I'm even running out of steam a little bit here. So then how would glycine interact to form a hydrogen bond? So to restate, the R group of threonine would be involved in this spring hydrogen bond. For glycine, remember, even though the side group can't hydrogen bond, there are parts of the backbone that could. So here's just in a single amino acid, but they would be involved in peptide bonds. And the oxygen and the nitrogen could potentially hydrogen bond. Why am I going into so much detail about this? Because if there's a mutation in a G protein in this region near the GTP binding site, which would you predict to be a mutation in which of these amino acids would cause altered GPCR signaling? One, both, or neither? So again, this gets back to what happens when you have a mutation. When you have a mutation, then let's assume that it's a, it still codes for an amino acid. It's just a different amino acid. So in the case, if you mutate threonine, you could change the side chain. So this would likely cause an inability to hydrogen bond with the electronegative oxygen. It's possible that parts of the backbone could interact still, but likely because of like the physical setup of this, they may not be accessible. So the threonine is most likely to cause a change in signaling if you mutate it. Whereas glycine, even it doesn't matter really what you change the R group to, because it will still, it initially formed a hydrogen bond with parts of the peptide backbone. Okay, so the hydrogen bonds are acting like springs that are holding. So here in what's shown here, here is the active state. In this state, the G protein can interact with downstream targets. When you cleave it, they flip out and are in a more relaxed conformation. But the relevant portions that bind with the effector proteins are now buried within, they're not accessible anymore. So probably the last detail that I'll go into today has to do with, there are things that are modulating the G protein activity itself. This is figure 3 in box 12-1. So the first thing to note is that the intrinsic GTPase activity of most G proteins is weak. What does that mean? It means that without an additional stimulatory factor, GPCR signaling would be on too much, because you would have GTP bound to the G protein. And remember, this is what can interact with downstream effectors. In beta adrenergic signaling, that was adenylyl cyclase. In this particular G protein, RAS, there's different pathways that we can talk about. It has other downstream effectors. So this is where you involve GTPase activator proteins, which are shown here in the figure. So the abbreviation is GAPS, and they help the hydrolysis occur faster. Damn it. Okay. So to help this GTP hydrolysis occur faster, this is where you have additional proteins called GAPs, GTPase activator proteins, that bind to the G protein and cause it to hydrolyze GTP more rapidly. So in the absence of these GAPs, or if there are mutations in these GAPs that negatively impact its function, the G protein will stay active for too long. So the binding of these GAPs basically make the timer run faster, so that you have a signal that's generated, but it's short. The second aspect of G protein modulation is the exchange of GDP for GTP. So the rates of exchange are also relatively slow. So this is the flip side of it, which is now we want to activate the signal rapidly. Without these GTP, GDP exchange factors, or GEFs, GDP, even though you have a conformational change in the G protein coupled receptor that's propagated all the way to the alpha subunit, it can't let go of the GDP quick enough unless you have these GEFs. So what is the consequence of that? The GEFs help downstream signals or secondary messengers be generated more rapidly in response to a signal. So you want to keep it nice, tight, and regulated. You have a signal, you have, I don't know, someone, the velociraptor is about to eat you. You need to have a signal be generated rapidly. Which modulator G proteins would be involved in that? That would be the GEFs. You have binding of epinephrine or something, right? And then you want to let go of GDP as soon as you can to make the active form, right? But now let's say you've run away, you've escaped the velociraptor. You no longer need G protein signaling. You no longer have ligand binding to the initial GPCR. How do you make sure that it's tight and the time it's self-regulated? That would be the GAPs that then help the GTP go back to GDP. Okay, so that pretty much sums it up for today. I'd encourage you, as I said before, to read box 12-1. I'm showing you here, there's a few paragraphs that I didn't really cover, but it gives you examples of, like, if you mutate a GEF or a gap, or the G protein itself, what effects might occur. In terms of the RAS G protein, mutations in RAS can cause cancer because one of the the biological process that it mediates isn't release of glycogen, but cell division, so cell proliferation. I just included, it's not really high resolution, but in case you wanted to see which parts of this box I'm talking about, like, I would encourage you to take a look at this, and be able to explain how mutations in these various factors, G proteins, the G protein regulators, the GPCR itself, vector proteins, downstream targets, all of these things, kind of similar to last week's in-class activity, be able to predict how they might change your activation of signaling, and justify why. On Wednesday, I will, like, very briefly in 10 minutes talk about the following subsections in 12.2. One, the idea of terminating the beta adrenergic response. We talked about two factors that were sort of involved in it, but there's one in particular, this process of desensitization. It's really the last theme of the logic of transduction pathways that we didn't talk about. So the idea that in the presence of a lot of epinephrine or ligand binding to your G protein coupled receptor, over time, there are actually active mechanisms to ensure that even though ligand is bound, the pathway won't be activated. This is akin to, like, having lots of exposure to light, but then over time needing a stronger input to have a signal be produced. Or, like, if you listen to something, your ears will get used to it. And then actually if you remove it, you actually hear a ringing. But anyways, and then I'll do a very brief overview of 12.3 because we need to pivot. To the best of my ability, I'm not going to cram everything in the metabolism chapters on you last second. I understand. Believe it or not, I'm on your side and I'm not trying to overwhelm you. But we do need to make a pivot to the metabolism chapters. So just a reminder of where we were and how far we've come. The big thing I wanted you to walk away with is to have a feel for the logic of signal transduction pathways. We hit a lot of these, certainly sensitivity, specificity, modularity, aspects of a localized response. Integration is really cool, and we won't have time to talk about it, but it involves multiple signals. So different branches of a pathway can be activated. We won't have time to talk about that, but we will talk about desensitization and adaptation on Wednesday for the arrestin. It involves phosphorylation of the GPCR and binding of this protein called arrestin. And yeah, I hope that you are beginning to feel more comfortable. You probably have been exposed to signaling pathways before, but hopefully you're getting a little bit more rigor in terms of the biochemistry underlying how signal transduction occurs. All right. Let me know if you have any questions on Moodle. Otherwise, I hope you all are doing well, and take care. Thanks.